Enzymes are protein catalysts that help biochemical reactions occur in our cells. Because of the unique shape of each enzyme, they have the capabilities to form and break chemical bonds by reacting to specific substrates. Substrates are any molecule that an enzyme interacts with. They bind to an active site, which is specific to the shape of a substrate and specifically the cofactor within the active site. The substrate and enzyme connection happens by an induced fit. This means that the enzyme can slightly alter its shape in order to fit a specific substrate. Once connected, this is called an enzyme-substrate complex. Enzymes don't change the level of free energy available in an interaction, but it does lower the activation energy, helping to reduce energy needed to start a chemical reaction. Organisms are so dependent on enzymes that without them, biological reactions, which are energetically expensive to begin with, would actually kill an organism if they didn't have enzymes. In other words, enzymes increase efficiency of biochemical reactions within cells. Degradation is an enzymatic reaction where a substrate binds to the enzyme and two or more products are made from the reaction. The result is usually energy being released. For example, a digestive enzyme lactase helps to break down lactose, a disaccharide found in dairy products. Please keep in mind that enzymes and substrates don't have these geometric shapes. These are there to help you understand enzyme activity. Lactose sugar binds with lactase at its active site. This causes a change in the shape to the lactase enzyme. Because of this change in shape, it literally breaks the glycosidic link that holds the lactose together. What it does is it forces the electrons to be so far from each other in a pair that they just break off, and the bond is gone. This causes lactose to be degraded into glucose and galactose, which are able to be used by cells. Notice the enzyme also goes back to its original shape, so it can be used again to break down more lactose. Interesting note, if you are lactose intolerant, it is because you don't produce enough lactase enzymes to digest lactose sugars. In synthesis, an enzyme forces a chemical bond between two molecules by bringing unpaired electrons closer to each other to help make a covalent bond. In the case of ATP synthase, it takes the molecules ADP and an inorganic phosphate and forces a phosphoanhydride bond, making ATP as a result. This happens with an insertion of food energy, causing the enzyme to change shape to make ATP. We will look more at the specifics of ATP production in Chapter 7. In reality, enzymes do not work alone, but in metabolic pathways. Each enzyme takes a substrate from a prior reaction and makes it into different products. The final product is what the cell needs. These pathways rely on enzymes functioning properly. Some medical issues stem from faulty enzymes not working in a pathway, leading to more or less product made than normal. And this is also how some pharmaceuticals work, either working for or against certain physiological processes either within you or within a pathogen causing disease. Enzyme A performs a synthesis reaction which binds to two substrates into a larger product. These products become the substrate for enzyme B, which changes the shape of the substrate. Finally, these products are degraded into two final products. There are many factors that affect enzyme activity. First, enzymes that function in catabolic reactions to degrade substances. The larger the substrate, the longer it will take to catabolize. Enzymes are proteins, and like all proteins, they can become denatured or lose their original shape if placed in the wrong environmental conditions. Colder temperatures slow down enzyme activity, therefore slowing down metabolism. You ever wonder why you refrigerate your foods? It's because bacteria rely on enzyme metabolic pathways to carry out life functions, but when they're exposed to colder temperatures, it takes longer for them to do those metabolic functions. And so therefore it extends the shelf life of your food before it begins to spoil. Warmer temperatures increase enzyme activity. One reason your body temperature increases when sick is to increase immune activity so that you can fight off disease much faster. However, if temperatures get too hot, they can actually denature enzymes. Pathogens, for example, exposed to fever for long enough can kill them since the enzyme activity is outside of normal temperature ranges, causing their enzymes to denature. However, if a fever gets too high or is not controlled, it can do the same for our own metabolic processes in our own cells. pH can also affect enzymes and denature them if exposed to a pH outside of normal ranges. In this chart, two enzymes, pepsin and trypsin, both target and break down peptide bonds in proteins found in our foods. Notice that pepsin functions best in low pH. That's because it's found in the stomach, where it's exposed to hydrochloric acid. Hydrochloric acid helps to destroy pathogens in our food. Once food enters the small intestine, however, the pH is neutralized by buffers made. Pepsin can no longer function at higher pHs, but trypsin, which also breaks down peptide bonds in proteins, can. Enzymes must be regulated based on the needs of the cells they operate in. Otherwise, too much or too little of products can be made for a given time frame. There's also outside forces that can inhibit enzyme activity against the instructions of the cell. One form of enzyme regulation is called competitive inhibition. This occurs when a similarly shaped molecule fits into the active site of an enzyme. In this case, the inhibitor and substrate needed to carry out an enzyme reaction are similar in structure, but the inhibitor reaches the active site first, and this typically shuts down enzyme activity. This is not necessarily bad. Perhaps the cell no longer needs more of the product and the enzyme needs to be turned off. However, we can use this competitive inhibition to our advantage. In bacteria, they create folic acid using metabolic pathways. Sulfa drugs are an antibiotic that mimics one of the substrates needed for an enzyme. When it works, the pathway shuts down. Bacteria don't get folic acid that they need, and the bacterium dies. Non-competitive inhibition occurs when a substrate binds to a site other than the active site, causing an enzyme to change its shape. Allosteric regulation is a reversible reaction that changes the shape of the active site, either activating or inhibiting enzyme activity. For example, let's say the enzyme displayed is part of the metabolic pathway used to make insulin. This protein gathers excess glucose in your bloodstream and stores it in the liver and muscles. Insulin production must stop after glucose levels are normal. So a regulatory molecule will bind to an allosteric site, which changes the shape of the active site, preventing the substrate needed in the production of insulin from working, thereby stopping insulin production. However, let's just say you eat a meal and your pancreatic cells pick up excess glucose in the bloodstream. The enzymes involved in the cell to help make insulin must now remove the regulatory molecule in order to activate the enzyme by giving it its original shape so the substrate can bind to it. Feedback inhibition is an important part of allosteric regulation, in which a reaction product made by a metabolic pathway can regulate and stop further production. Feedback inhibition is important for preventing waste of necessary substrates and products, stopping activity when an organism no longer has the necessary nutrients and energy to power that reaction, to help prevent higher doses of end products that could be toxic in large amounts, but mostly feedback inhibition works to maintain the homeostasis of an organism's physiology. So here is a normal metabolic pathway. Let's say that organism, at the present moment, does not need any more of a given product. It can send one of its end products to enzyme A. This causes enzyme A to change shape, preventing further production. If the organism needs this product again, the substrate is removed and the enzyme goes back to its normal shape. Enzymes can't work without helper molecules. Many of these are essential nutrients you need in your diet. Two types of helper molecules are cofactors, which are inorganic ions like magnesium and iron. DNA polymerase, an enzyme that builds DNA, requires zinc ions as cofactors to help function. Coenzymes are organic molecules and are mostly vitamins. Vitamin C can be used to build collagen to help strengthen tissues throughout the body, making vitamin C an important coenzyme. Because eukaryotic cells are compartmentalized into organelles, they utilize different enzymes based on the internal fluid of the organelles as well as the organelle's main function. For example, lysozyme enzymes function in digestion, while mitochondrial enzymes function in cellular respiration, which is the topic of our next chapter, making ATP. And now, so in our next chapter, get ready for the mighty mitochondrion.